starting from pioneering studies conducted in the early 1900s. The cell culture process has evolved into one of the most widely used experimental methods for scientific research. Cultivating cells is a complex procedure that demands precise and controlled conditions. Pressure, temperature, and pH levels among them. A significant role is played by the gas mixture flowing through the culture and its controlled gas flow meter. Instruments for managing gas mixtures must ensure high precision throughout the entirety of an experiment, demanding accuracy, stability, and repeatability. Equally important is the capability to work with an instrument capable of dynamically adjusting gas mixtures. This adaptability is crucial, as changes in conditions for specific cell types can result in shifts in gene, protein, and enzyme expression. One pivotal aspect of in vitro cell growth is maintaining complete control over parameters throughout the process. While cell culture applications may greatly differ, the growth medium stands as one of the most critical of these parameters. Typically buffered with sodium bicarbonate or carbonic acid, the medium's pH value must be precisely constant for proper cell growth. CO2 content in the culture atmosphere directly influences medium pH, making control over the gas mixture in which cells grow yet another fundamental process parameter. Due to air's low CO2 content, less than 0.04%, it's not a viable option for cell culture. In most common applications, cells grow in an atmosphere of 5 to 10% CO2 within specialized CO2 incubators. Basic CO2 incubators use pre-mixed gas cylinders, wherein the user cannot adjust the amount of CO2 in the flow. More advanced incubators include CO2 sensors for feedback regulation, though they are less common, and more expensive than basic models. Working with static gas mixtures featuring fixed or adjustable CO2 amounts now forms the foundation of cell culture applications. Over the past 2 decades, however, successful experiments have been conducted using dynamically obtained gas mixtures. There is an increasing demand for instruments capable of controlling both O2 and CO2 proportions in the cell culture atmosphere. Fine control over gas mixtures facilitates experiments requiring induced hypoxia, hyperoxia, or hypercapnia states. Dry air typically contains approximately 78.08% nitrogen, 20.95% oxygen, 0.92% argon, 0.04% carbon dioxide, and other trace components, largely noble gases. A hypoxic gas mixture has a lower oxygen content than air. The oxygen level in a hypoxic mixture ranges from a minimum of 0%, anoxia, to a maximum of 21%. Conversely, gas mixtures exceeding 21% oxygen are considered hyperoxic. Inducing hypoxic conditions in cell cultures is a common technique for studying cardiovascular diseases, tumor research, apoptosis, gene expression, and enzyme studies. Hyperoxic conditions are typically induced for lung injury studies. Hypercapnic mixtures vary CO2 concentration instead of O2. These mixtures contain 21% oxygen, and an increased CO2 amount compared to air. Cell culture studies in hypercapnic conditions collect data about ischemia. Often, a combination of hypoxic and hypercapnic mixtures is used in various experiments. Basic and advanced CO2 incubators serve standard cell culture applications well but fall short when dynamic gas mixtures are required. Furthermore, neither type of incubator allows control over O2 levels in the culture atmosphere. For applications necessitating dynamic gas mixture preparation, gas flow meter capabilities, and comprehensive control over mixture composition, MCQ Instruments recommends using its gas mixer series. This gas mixer is designed to manage 3 components of dynamic gas mixtures, offering versatility and adaptability for various lab applications. The gas mixers are calibrated with native gases. For cell culture applications, the standard configuration involves N2 as the balance gas and O2 and CO2 as the solute gases. Upon request, the instrument can be set with other desired non-aggressive gas media. For each channel, these instruments guarantees high accuracy, 1.0% of set point, high repeatability, 0.16% of reading value, and rapid response time for set point value changes, 50 milliseconds. Accompanying the instruments is the Gas Mixture Creator software for gas mixture management. Usable on common desktops, laptops, or touch screens, the software empowers users to take full control of the instrument and its functions with complete automation. Start now using these powerful devices and increase the pace of your cell culture experiments. Thanks for watching.